Kind of.
What? What are they doing? Do you have any idea? Actually, it is very difficult from this face-to-face class to move to online because the format is different. We have a lot of things to discuss there, but in online, that already there video recordings and everything is there. We don't have any time to change the format. We are in almost like war situation. Like that. Within 2 days, we have to move everything. No. I have a plan that probably I will keep this time. So just like what we have the regular time, like 11 to 12:20, but we'll move to. I am trying either WaveX or the Canvas conference. Okay. So using the Canvas conference, we can have a meeting during this time, and it will be just like the regular classes. You can ask question, I can talk, and we can do the whiteboard to draw all those things and everything there. So it will be almost similar like the regular classes, but we'll have no contact with each other. Now the rule is that I have a lab, and the problem is that there you have to maintain 6 feet distance, like gel spectrophotometry. Can we do? And we are generally doing in groups. It is not possible. So
It's hard to be in a group 6 feet apart.
yeah, it is not possible. The 6 feet means this kind of distance. How can I maintain all the time
We
with the
just
students?
we had to yell at each
And
other.
each and everybody need to maintain that thing. So we are in almost war situation. I don't know. Your homeworks and everything is with me. So what I will do when we are situated with this situation a little bit, I will just scan those copies and I'll send it back to you. Okay. And all those homeworks and everything, it will be online. So you can just for this spring break, I will not give you any homework. Just enjoy your spring break. And after that, we have a little bit more homeworks there. And particularly, I need to go for this class actually, a little bit homework. So then we'll do just either email or through Canvas. I'm trying to maintain in Canvas so we can have in one area and we can maintain everything there. So I'm trying to do that now. And check your internet connection and everything there because this is very, very important. And that's why I need the email address and everything. That's why I send you the emails. So those are the things we need to do right now. And let's see what will happen. Probably I'll because when I'm checking the rest of the syllabus, we have a lot of interesting chapters and really discussing chapters, there, particularly the gene mutation, molecular genetics, and biotechnology. These needs a lot of discussion. Otherwise, you can't understand all these things. Particularly, gene mutation means joining and those replacing all those DNA segments. Those are really things. And next, we have the genomics part. So genomics part means already the nucleotide analysis and matching with those nucleotides, haplotype, all those things there. So there are a lot of discussions there. Let's see. But one thing is that if anybody interested to volunteer with me, so I just want to test that whether this system is working or not. Certain for when I will make these things a little bit okay, that okay, all those courses and there, everything, what it is working. I just need to test with somebody that it is really working, that I can use the whiteboard. So I'll send you one email. If you are interested, we can set up one time just for 10-15 minutes. Then we can see that whether you can see, because I cannot see that one. That whether you can see the PowerPoints, whether you can see the whiteboard and what is the screen things. That is technical part. We'll do in one time. Just after spring break, before the class, something like that. We'll do that. Okay. And another thing is that we got a new notice that we will resume the class on April 30th. So it is like that spring break and 2 weeks after spring break. I am calling this one as quarantine period. So we will be online for this period. But we got a notice that there is a chance that we cannot come back. Okay, so be prepared for that. In such case, everything will be online, and generally I'm doing the quiz and test online, so there will be no problem for you. We can finish the course. It is not a problem. Okay, so today we are going to talk about another very important thing, that is the regulation of gene expression. Like we were talking about this one before, that we have cell everywhere inside our body, in our eye, lips, skin, liver. There is everywhere there is a same cell with the same number of chromosomes, same number of genes, but they are different. Our eye cells are different than our skin cells. Why? Okay. Or the liver is doing different kind of function rather than the kidney. Why? Because everything is gene controlled. We know that everything is gene controlled, and there is the expression of gene means that is the proteins. Okay. So each and every gene is synthesizing some kind of proteins. So each and every cell theoretically have the same kind of proteins, but it is not. What is happening there? That means there is a chance that some genes are acting in one cell, one kind of cell. Some genes are not acting in other kind of cells. Okay, so this is called gene regulation. It is the switch on, switch off process. When it is required, the gene is acting. When it is not required, the gene is not acting. Okay. Suppose in our eye cells, some genes are there which can regulate these pupil and all those functions of the eye. Those genes are activated on eye cells, but those are not activated in our skin cells because our skin has different functions. But the cell is same, the chromosome number is same, the genes are same, but their proteins are not same. Their expression is different. That is called the regulation of gene expression. That is a switch on, switch off process. When it is required, the gene is switched on or switched off. Okay? Switched on and switched off means that is another thing, that either the system is off, but when it is required, I will switch on the system. Okay, so there are two ways. And to discuss this pattern, we need to know about a couple of structural areas. That is, if this is a gene, there are certain areas. This part, suppose, is called structural gene. What is the function of this structural gene? These two, suppose, are structural gene. What is the function of structural gene? They can produce the product. Suppose we need some kind of enzyme. These genes, they will produce the product. Now the question is that during transcription and translation, we were talking about one structural part with a promoter. Okay, but it can happen that there is a promoter, but the whole promoter is for a couple of genes. Okay, there are several genes there, but there is only one promoter. It is regulating all those genes. So from each and every gene, there will be one synthesis of one different kind of proteins, but only one promoter is regulating them. Here we will talk about in that part. So we will have a promoter region. Okay. So what is the function of promoter region? That it will allow RNA polymerase to bind with this part, then go for the transcription of this part, and the product will be produced. mRNAs will be produced. One mRNA from here, one mRNA from here, and there will be translation, and the proteins will be produced. Okay. So that is called the structural genes, and there is another part here. This is called operator. This portion, it will operate. So RNA polymerase can bind with this promoter part, but this operator part is the doorkeeper. I will allow you to enter, or I will not allow you to enter. Okay. Suppose I am outside. One person is there. He is telling me that you can enter into this classroom or you cannot enter. That is the function of operator. Whether I am there or not, that doesn't matter. But it will say that whether you will enter into the structural genes or you will not enter into the structural genes. That is called the regulatory genes, regulatory elements actually. And after that, there is some upstream of here. There is some upstream of here, a little bit upstream. There is another part called the I gene. It is producing one product, that is the regulatory gene, producing one product which is called inducer or repressor. One kind of protein is called inducer or repressor. Actually, this protein acts with operator region to say whether the RNA polymerase will go for the structural gene or not. Okay. So regulatory genes encoding the products that interact with other sequences. That means the inducer or repressor, they will create one product that interact with other sequences, that is the operator, to run these things or not. That is called the regulatory gene. And regulatory elements that are not transcribed but play a role. This portion is not transcribed. This portion is not transcribed but playing a role. Okay, whether the RNA polymerase will bind or whether it will move or not. Now, based on this one, we have a couple of things as we were talking. That one, the system is on, but we need to switch off the system. Or the system is off, we need to switch on the system. Okay, so one thing is that constitutive expression, that is continuously expressed under normal condition. If the condition is normal, it will create the protein synthesis and everything, it is going on. Another is called positive control. Positive control means if the protein is not there, then the system is on. If the protein is there, it requires to do something, then it increases the synthesis. That is called the positive control. And negative control is that when the protein is there, it does not require any kind of other things, then it will switch off the process, inhibit the whole process. That is called the negative control. And to describe all those things, we have this operon model. Operon model in E. coli bacteria, and it is a good example of this gene regulation, but there are several other models, and in eukaryotes, the model is a little bit different. Okay, this is one example, but that doesn't mean that everywhere our iCell is doing the same kind of things. There are several other things there, but this is one example. It is called the operon structure. The operon means this whole part, one inducer gene, one promoter, one operator, and structural genes. This whole system is called the operon structure. And there are two very important operons we need to know. That is, one is lac operon and another is TRP operon. Lac operon means lactose operon model, and TRP operon is tryptophan operon model. So this is the operon structure. Here is the regulator gene that is producing either repressor or inducer. Repressor means which can switch off the process. Inducer means which can switch on the process. One similar kind of protein, but the function is different. Okay, that is produced by this regulator gene. Okay. Regulator gene must have one kind of promoter. It is a total different sequence. They can have one promoter, but that is different. Okay, so they can produce one product by transcription and transport. This regulator protein, it can bind with the operator. Okay, what is the function of this promoter? Promoter just calls the RNA polymerase. Come here, sit here, and do the synthesis. That is the function of promoter. I am giving you space to bind with me and you can synthesize here. Now operator is controlling that whether this RNA polymerase will move or not, and that is controlled by this protein. If it will bind with the operator, In such case, if it is a repressible, then it will block the synthesis. If it is inducible, then it will start the synthesis. Okay? What is the function of these structural genes? There are several structural genes are there, and each and every structural gene can create or synthesize one kind of protein. Okay? This whole structure is called the operon structure. There are mainly three parts: the promoter, operator, and structural genes. And another thing is that the inducer protein. Okay? So negative and positive control, inducible operation, that is transcription is usually off and need to turn on. That is inducible. We need to induce the transcription. Repressible means transcription is usually on. We need to switch off the process. That is called the repressible. Now, what is negative inducible operands? The control is negative, and which molecule is binding to the operator is generally inhibiting the transcription. So, the molecule we will call the repressor that will bind to the operator and can block the RNA polymerase to move. So, that is generally the function. So generally, this is off. The whole system is off. In normal condition, the inducer is producing or repressor molecule, it is producing the repressor molecule. Repressor molecule is binding to the operator and the whole synthesis is off. This is the normal condition. And what is the function of this molecule? It needs to be turned on. Then the protein is required, that is the inducer. Okay. So no inducer is present here. The first picture is that there is no inducer. Inducer means some other molecules. Okay. In such case, the repressor protein is binding to the operator and stopping the transcription process. When the inducer protein is there, here, it is binding with this repressor, making is inactive. So it cannot bind with the operator. In such case, the synthesis is on. Clear this part? These repressor molecule in normal condition can bind to the operator. In such case, it will not allow RNA polymerase to move to the structural genes. So there will be no synthesis of proteins. When the inducer molecule is there, some kind of precursor, some kind of things. In the Lac-Operon, we will know which is inducer actually. It will bind to this repressor. Actually, it is functioning with the repressor molecule, making it little bit distorted that it cannot bind with the operator. In such case, it is on. This is inducible operon. So it was switched off and the inducer is turning it on in this way. In this case, the transcription is on. In normal condition, the transcription is on. But when that molecule, that is a repressor molecule, is present, then it is switched off. In such case, if it is negative control, that is using that repressor molecule to bind with this operon region, that means in first case, it cannot bind with the operator. When the molecule is present, then it can bind to the operator. Does it seem like that? Think so. In normal case, it cannot bind with the operator. When this repressor molecule is present, now we can call it repressor because it is repressing the transcription. When the repressor molecule is present, then it can bind to the operator and can stop the synthesis. So that one was off. That molecule, inducer molecule, is creating some kind of distortion to that thing, and It cannot bind to the operator, the switch is on. Here, normally it cannot bind, but that molecule is creating again the distortion to this molecule, so it can bind to the operator and the switch is off. And this positive one is that, suppose this is the negative one, that switch on, switch off. But positive one is that when the molecule is present, suppose glucose is present, to synthesize glucose, or glucose is not there, but we need glucose. In such case, we need to run the operon model. That is called the positive synthesis or positive inducible operon. I will give you the example later. At first, this negative operon. This example is lac operon. In lac operon, there are a couple of genes. That is a lac-I repressor encoding genes. This is this one. And as it is lac operon, we call this as lac-I. Okay. Then lac-P, the promoter. This one. Lac-O, the operator gene. And the structural genes are there. This is Z and Y and A. Three structural genes. And they are synthesizing three different proteins. The first one is beta galactosidase, next one is permease, and the last one is transacetylase. These three enzymes. And what is the function of these enzymes? So, if you put E. coli in the lactose containing medium, suppose lactose means where is lactose? In milk. Normally in milk, the lactose is there. So if you put the E. coli in the lactose, there is a lactose in this medium. They need to synthesize that lactose inside their body. What is the function there? That permease is helping that lactose molecule to come inside the cell. That is the function of permease. So if the lactose is there outside the medium, they need the permease, okay? Because it will allow lactose to enter inside the cell. When lactose is entering inside the cell, then beta galactosidase, that is the main enzyme, it can convert the lactose into glucose and galactose. Lactose is disaccharide, okay? And glucose and galactose, what is this? Monosaccharides. Monosaccharides means what? One carbon, two carbon, one carbon atom, one carbon molecule. Are you sure? One carbon molecule. What is the formula of glucose? C6H12O6. That means 6 carbon molecule. 6 carbon molecule. Monosaccharide means 1 unit. Okay? Monosaccharide means 1 unit. Either it could be, glucose is called hexose. Hexose means having 6 carbon molecules. Ribose is called pentose. Deoxyribose is called pentose, having 5 carbon atoms there. Those are called pentose. But those, all of them are monosaccharides. Okay? When two monosaccharides joins together, they will form the disaccharide. And that structure is C12H22O11, not C12H24O12. One molecule of water is coming out from there. Okay? That is called the disaccharide. So lactose is disaccharide. What are other disaccharides? Other than lactose? What is another monosaccharide? We have two monosaccharides here, glucose and galactose. What is another monosaccharide? Glucose, galactose, and fructose. Glucose, galactose, and fructose. And what are the disaccharides? One is lactose, another is maltose, another, which is very common for our everyday life. This is very common in our everyday life. Sucrose, sugar, table sugar. That is the sucrose. Sucrose is made up of glucose and fructose, that is the sucrose. Glucose and galactose make the lactose, and glucose make maltose. Okay? So these are disaccharides. Another term is called a polysaccharide. What is polysaccharide?
Many units
Many units of what?
of carbon compounds or
Not
units
carbon
of
components. Many units of monosaccharides, but modified form. So generally monosaccharides are C6H12 O6, and in case of fructose, that is maltose, sucrose, and that is lactose, we can have C12H22O11 with the release of one molecule of water. Okay. In case of polysaccharide, it is C6 or C5H10 and O5, or C6H10O5, generally C6H10O5. That is the main structure, but it is in several numbers. This N could be 10 to 1000, even more than 1000. A long chain of that. From each monosaccharide, one molecule of water is coming out. So it is more stable, and the long chain of that one, that is called a polysaccharide, which is, what are the polysaccharides? We cannot digest.
cellulose.
Cellulose and
Does it start with a G?
no, start with S. And one is starting with S, another is starting with C. One C is there, cellulose. What is another one? We cannot digest, but the cow can digest because they have cecum, and our cecum is now appendix. We don't have that kind of bacteria in our body, so the herbivores can digest that, but we cannot. These are some general. Yeah, starts with this. Right. Starch.
Ah.
And another is C, chitin. Chitin is used to actually for our operation. Any kind of surgery, the chitin threads are used to heal up that kind of things, to suture those kind of areas, so it can be digested later and there will be no problem. We need not to take it out. And chitin is actually present in any kind of exoskeleton of the animals. Exoskeleton means some kind like in insects. They can mold from pupa to normal things. That pupa is made up of little bit of chitin. So we generally take out chitin from there. So these are the things. Anyway, let's come to this part. So when there is milk there, the lactose is there, and if you put the E. coli needs to digest that lactose. So it needs generally three enzymes. One is permease, one is beta-galactosidase, and another is transacetylase. Permease is helping that lactose molecule to come inside the cell. Beta-galactosidase has two functions. One function is that breaking down that lactose into glucose and galactose. That is one part. Or it can transfer this lactose into allolactose. This allolactose again is broken down into glucose and galactose. This is the function of beta-galactosidase. So the end product is glucose and galactose, which is required for their E. coli body. Okay. So now the question is that when the lactose is present, they need to synthesize permease, beta-galactosidase, and transacetylase, right? The question is that when the lactose is not there, they don't need those enzymes. Okay. So in such case, when the lactose is not present, then that operon is off. We don't need any kind of beta galactosidase, we don't need any kind of permease, we don't need transacetase. When lactose is not present, absence of lactose, then this regulator protein that is called the repressor, it is binding with the operator region and can stop the synthesis of the whole structural genes. There will be no production or no synthesis of those enzymes. When lactose is present, then we need to turn on this whole thing. We need all those enzymes. Okay. When lactose is present, then it is forming allolactose. That allolactose is binding with the repressor, making it inactive. So it cannot bind to the operator. So the function is that production of allolactose. That allolactose is binding with the operator, that is repressor protein. Repressor protein cannot bind with the operator. In such case, RNA polymerase can move. If RNA polymerase can move, then they can form this beta galactosidase, permease, and transacetylase, which is required for lactose synthesis. Clear? This part? But they have certain mutations there. Couple of mutations. Mutations for each and every one. This inducer, they have mutation. This promoter, operator, Z, Y, A, all these genes, they have certain kind of mutations. I'll go here very little bit about this one. So, regulator gene mutations, that means either I minus or the I plus. And I plus is dominant over I minus. If there is I plus, that means the repressor is active repressor. If this is I minus, then it is inactive repressor or non-functional repressor. Let's see the structure. Now, the function of repressor is that this molecule, it can act in any of the two chromosomes. Okay. It is actually producing one kind of protein. That protein can bind to the any of the strands. This one, at first think about the absence of lactose. There is no lactose present. There is one lac I minus gene, and it is creating the non-functional inducer or repressor. But the normal one can produce the functional repressor. Generally, the mutation is not on to both strands of DNA, only in one strand. So that repressor can work on both the strands of DNA. Okay. If there is no lactose, the function of the repressor is that binding with the operator and stop the synthesis. But only one functional repressor can bind to both the operator. That means there are a lot of proteins, a lot of molecules there. They can bind any one of the strands and can stop both synthesis of both the strands. When lactose is present, then the same thing, whether it can produce active or inactive repressor, that doesn't matter. One active repressor is there. It can bind with the lactose or allolactose, can form the inactive repressor, then the synthesis is on for both the genes. Clear? Next, if it is a super repressor, there is another function here. So here is for I, we have I plus, I minus, and IS. IS is called super repressor. And that super repressor cannot be activated or deactivated by lactose or anything. That is super repressor, Superman, something like that. It cannot be distorted by those things. When super repressor is present, it can stop the synthesis by any way. Okay? So when IS gene is there, there will be no synthesis in the lac operator. In case of operator, there are two mutations. One is called OC and one is O plus. There are two things. OC is dominant over O plus, and this is cis acting. Cis acting means that means it is acting on the same strand. So it was transacting. That means whatever the protein is there, that protein can act on either of the strands. But it is cis acting, but that means it is only working on this strand. Okay, either it is C or it is O plus. So if it is C, C means that cannot bind the repressor anyway. Okay, so if the C is here, whether we have the active repressor or whether we have the inactive repressor, that doesn't matter. Okay, because it cannot bind that repressor. Then the synthesis is on. Clear? Then we have the synthesis of promoter, that is P minus and P plus. That is also cis-acting. That means on the same strand. If P plus is there, then it can bind the RNA polymerase. If P minus is there, then it cannot bind the RNA polymerase. So suppose in such case, if it will happen that we have I plus here, we have P minus here, we have O plus here, C plus, Y plus, and A plus. All these structural genes have this kind of mutations. Okay. If the condition is like that, then what will happen? Lactose is present. What will happen? Lactose is present. We have this construction. What will happen?
It fails to produce galactosidase.
Beta-galactosidase. Why? What is the explanation here?
Is it
Go step by step. So I plus gene is here. So from I plus we can have the repressor, active repressor. Lactose is present. Lactose will bind to this repressor, make it inactive. If it is inactive, then it cannot bind to operator. O plus is here, so operator is on. It cannot bind with the operator. Okay, that means the system is on. But P is minus. RNA polymerase cannot bind to this site. What will happen in such case? This part is on, but P is minus. RNA polymerase cannot bind with that part. What will happen? Will there be any synthesis or not?
they wouldn't be able to synthesize the lactose?
There will be no synthesis because RNA polymerase cannot bind to that site. Okay. If it is plus, if it is also plus, and if it is C minus, what will happen? Lactose is present. And go step by step. What will happen? First step is that lactose is present. It can bind with the repressor. Repressor will be inactive. That means the system is on. The first conclusion. Okay. Operator region is plus. That means there is no problem for binding with the operator. Operator is fine, so the system is on. P plus is present. That means RNA polymerase can bind with the promoter site, so the synthesis is on. But Z minus is there. It cannot produce any beta galactosidase. So what will be the end product of this system? Only permease and transacetylase. There will be no beta galactosidase. Clear? These are a couple of things, and there are a lot of problems with that, these mutations and those things. You need to find out in this way. Now, here the lactose is present. If the lactose is absent, what will happen? In this system, minus prime, what will happen? They can go step by step, each and every step. If you do this kind of things for one or two, three problems, then it is very okay, easy for you.
I thought if lactose isn't present, the repressor keeps the enzymes from functioning.
So repressor will bind to this operator and the system will be off. Whether the promoter is there or not, that doesn't matter. Whether the Z minus gene is there or not, that doesn't matter. Repressor will bind to the operator and the system is off. Okay? If this is OC, what is that? Again, think about this. Lactose is absent and the operator is OC. What is that? What is the function of OC? Cannot bind the repressor. So repressor is active here because the lactose is not present. Repressor is active. But OC is there, so repressor cannot bind to this one. So the system is on. As P plus is there, RNA polymerase can bind here. The system is on. Here is G minus. For Z minus, there will be no production of beta galactosidase. Only permease and transacetylase will be produced from here. These are the steps you need to think about that. Now, in such case, if it is P minus, what will happen? Lactose is not present. This is P minus, this is OC.
Will it not connect? Like, will the repressor not connect either one?
Not connect. This is the repression. Lactose is not present means repressor is active. OC is there. Repressor cannot bind with the OC. For that reason, as OC is there, that means the system is on. P minus is there. RNA polymerase cannot bind with the P minus. So the whole system will not work. Okay. Now, if this is IS, what is that?
It won't work.
Because for OC it will not work, though it is super repressor. Okay. If O is normal, then anyway it will stop the synthesis because it is super repressor. So So these are a couple of things you need to think about that one, and that is the main thing of lack of plan. Okay. We have a couple of problems. Later, after spring break, probably I will give you those kind of problems to understand this whole thing. Okay. Now the positive control. Positive control means here the thing is that just go to this thing. When glucose is low, it is working on the lactoperon also. Because in the lactoperon, we saw that beta galactosidase, the function of beta galactosidase is the breaking of lactose into glucose and galactose. When in the cell the glucose is low, this lactoperon is working in some other way. Okay, the same thing, but it is working in some other way, not in that way. That is that when the glucose is low, the level of CAMP, cyclic AMP, is very high. Okay, that cyclic AMP is binding with the cap protein, and the cap protein actually enhancing the whole thing to synthesize for the glucose. That means produce these three enzymes, break down the lactose, and produce the glucose and galactose. Okay. So this is enhancing. When the glucose level is low, then but the lactose is there inside the cell, then it is enhancing the whole system to synthesize more glucose. When the glucose level is high, they don't need any more glucose. When the glucose level is high, then the cyclic AMP level is low. This is controlling, the glucose level is controlling the cyclic AMP. There is some other biochemical pathway that whether it is controlled by in this way. Glucose level is high, then CMP level is very low. It cannot bind with the cap, then it cannot synthesize or enhance the whole thing. Okay? This is called the positive control of lactoperon. Now, one is called the negative repressible operon. Negative repressible operon means when tryptophan level, this is called the TRP operon or tryptophan operon. Okay? When the tryptophan is low, they need more tryptophan, more TRP. And the TRP is required by the synthesis, that is also a biochemical process, and there are 5 enzymes required to synthesize the tryptophan from what is meant. Okay, that is present in, it is coming from outside the body, that is from food materials or other things. They need to produce this tryptophan. What is tryptophan? Tryptophan is one kind of amino acid. Okay, they need to break down the other one to produce the tryptophan. Okay. In such case, when the tryptophan level is low, then the repressor, what is producing here, that cannot bind to the operator, then the system is on. Okay? So they can break down this coenzyme to produce tryptophan. When the tryptophan level is high and it is continuous, that system is off, the tryptophan is producing. When the tryptophan level is high, they need to stop the system. When the tryptophan level is high, that tryptophan molecule binds to the repressor, make it active, then it can bind to the operator and can stop the synthesis. The same thing, that is there it was lactose and here is tryptophan. Lactose was controlling the repressor not to bind. Tryptophan is acting the repressor to bind. This is the difference between these two. Okay, so when lactose is present, then we need to turn on the system. When the tryptophan is present, we need to turn off the system. Clear? But in tryptophan operon, there is another part which is called the attenuation. What is attenuation? Attenuation is like that there are TRP codons here, but this whole mRNA that can make some kind of structures. Okay, there are 4 regions here: 1, 2, 3, and 4. And these 4 regions can bind with each other. Okay, when the tryptophan level is high, then we need to stop the transcription. Okay, In such case, what is happening? 1 binds with 2, 3 binds with 4, and can make this kind of structure, then the synthesis is stopped. Okay? In case of other way, when the tryptophan level is low, then these 2 and 3 can bind, 1 is open, 4 is open. In such case, the synthesis is on. So just like hairpin structure, once it is binding with this way, then the level is stopped. Another time it is binding in this way, then the synthesis is on. Just the change in their structure. This is called attenuation. And I put these 2 slides here that is showing that the dynamic model of this attenuation. You can just go through this. Okay, now we need to talk about our eukaryotic system. Okay, in eukaryotic system there are a lot. One main important part is the GAL4 system in yeast. GAL4. Okay, GAL4 system is a little bit complicated. It is in your book. You can read that one if you are interested. But we will talk about some little bit of thing here. One is DNase1 hypersensitivity. We already talked about that one in our previous system, that there are some parts which are sensitive to DNase1, and those things, when DNase1 is there is no synthesis there. Okay. The next very important part is the histone modification. So in our chromosome, eukaryotic chromosome, we have histone proteins. And histone proteins, the DNA is coiled onto histone proteins. Okay. So generally, there is no such open space for transcription. Like this structure. There is no such open space for transcription. Only the linker DNA is there. If the promoter is within this region, this coiled region, what will happen? Will there be no synthesis? There is synthesis. But how is it happening here? That is the chromatin remodeling. What is that? This is very simple. The promoter region is under some coil. What is happening? They can just move the histone protein so it can open up. Just moving the histone protein, so a couple of coils later, keeping this one open, this DNA part is open, then the synthesis can be done. This is called histone modification. Okay. And this histone modification can be done either that way with the histone modification proteins or chromatin remodeling complex that is made up of couple of proteins, almost like 10, 12 proteins are there. Okay. And they can make this kind of modification. When the synthesis is done, they can come back to the original state. Okay. This is always happening in eukaryotic chromosome because eukaryotic chromosome is not open. Another is called, we know the histone has some kind of tails. Histone tails. We talked about this one, the histone tails. The histone tails has one factor that is called the acetylation. What is happening there? In histone proteins, they have long tails like this. What is the function of histone tails? What is the function of histone tail? Positively charged histone tails can help the negatively charged DNA to bind with the histones. Okay, that is the function of histone tail. What is happening here, when there is acetylation in these histone tails, it becomes reduced. Okay, the length of the histone tail becomes reduced due to acetylation. In such case, it can open up the DNA. It can open up the DNA for the synthesis of protein. Okay. And the alternative splicing. What is alternative splicing? Just tell me. I'll not tell you. Explain this. Alternative splicing. What is that? What is alternative splicing? What do you think? Yeah. What is RNA splicing? What is RNA splicing?
Like the enzymes will start and stop at certain spots on the mRNA, and then it might skip like a few spots. And then
Doesn't it cut out part of the gene?
Cut out part of the gene. Cut out part of the gene means what? Cut out part of the mRNA. What are the cutout parts?
These parts that don't need to translate.
They don't need to translate those parts. What are those parts called? Mutation? Mutation is some different thing. It is in normal.
Or do they split out the portions like the beginner?
What are those parts? mRNA is produced, it is containing some other parts. You need to know these things. Already we have done with the transcription, post-transcriptional modification, RNA splicing, we have done with that. And these are the questions. What is the difference between pre-mRNA and mature mRNA? What is the difference between these two? Pre-mRNA and mature mRNA? Parts of the introns. So those are called introns. And which is required for protein synthesis? What are those? Exons. Okay. And what is alternative splicing? Okay. We talked about that on the 3' end and a lot of things are there. This is mRNA. We have exon, intron, exon, intron, exon, like this. Okay. So during splicing, what will happen in normal case? E1, E2, and E3. I1 and I2 will cut out. This is the normal splicing. In such case, it can produce a kind of protein. Okay. What is the difference between mature mRNA and pre-mRNA? There will be no introns in the mature mRNA. In the pre-mRNA, there will be introns. Okay. Now, what is alternative splicing? Alternative splicing means the introns are not like that every time the introns will cut out. Okay. In such case, if the splicing is something like that, E1, I1, E2, there is a cut here. It will create a different kind of protein. Okay. Because some of the introns part are into that is overlapping part with the exons. Okay. Or the splicing could be like this. Only the E1 and E2 will be there will be no E3. Splicing could be like this, only the E1 and E3 are here, there will be no E2. This whole part is spliced out. Okay. In such case, what will be the end product? This will create one kind of protein. This will create one kind of protein. This will create one kind of protein. And even if possible, this will create another kind of protein. Same structure, but the different kind of cutting. And if it is overlapping parts, sometimes it is cut out here. Okay. We have that overlapping part in the introns and exons. Each and every one will create a different kind of protein. That is called alternative splicing. So based on the splice site, they can create different kind of proteins because we have 21,000 genes and we have 150,000 proteins. So one gene can produce various kind of proteins, or one mRNA in such case can produce various kind of proteins. In such way, this is called alternative splicing. This is another gene regulation part. When it is required, this kind of protein will be produced. When it is required, this kind of protein will be produced. The same mRNA, but the proteins will be different due to this kind of splicing. This is another gene regulation. Okay. So we have various kind of things. In eukaryotes, we have a lot. These are a couple of examples. And for your course, I think this is quite enough. Just, if you are interested, just read the GAL4 part, because GAL4 is a little bit complex, okay? And you can just read the GAL4 part. Otherwise, the alternative splicing, histone modification, and DNase 1 sensitivity, these three are the main part for eukaryotic gene regulation. Okay? For prokaryotic, tryptophan attenuation, TRP operon, and lac operon. And I will give you later the couple of problems on lac operon. Okay? So go through the lac operon. And these two parts I will not go too much details. I think it is quite enough. Do you think? Or you want to go more? Okay, that's fine. Okay, so we are done for today. I think so. I made it these two chapters to be solved. I told you that these two chapters I will make solved. Later we'll go for the mutation part, then the biotechnology part. It will be online. Anyway, those two are very, very important. There are a lot of things there. I probably biotechnology part I will increase a little bit more, and the immunogenetics part I will make it a little bit soft. But immunogenetics is a little bit critical. Okay? Immunogenetics and developmental genetics. Because developmental genetics is also a little bit. And rather I will go for the population genetics a little bit. Okay? So, any question about the previous chapters? That is, I need to go a little bit about those things like DNA replication, transcription, and translation. Because since those chapters, I saw your faces are a little bit different for the molecular biology classes. I know the replication transcription is a little bit different to understand. But if you have any question, just let me know. So when I will go for the biotechnology classes, I will go for the replication part a little bit more, because the synthesis of that is what we will do there. We'll tag the DNA with different biochemical markers, different fluorescent markers. Suppose we need to find out the telomere region in the DNA. Where is the telomere region? Okay. We need to prepare the short fragment of DNA that is prepared in the lab. We can attach a fluorescent dye into that one. Then we can put into the cell to make the replication there. During replication, that part of the DNA will attach to the original DNA with the complementary sequence. Then we can find out, okay, this is our actual part. And the polymerase chain reaction, that is called the PCR. Using the PCR, we can make multiple copies of DNA in the testing. And generally in lab, we are using that one. I don't know whether you did it or not. Did you do the DNA gel? Did you do DNA gel? During DNA gel, what did you do? You know? What is
I
it?
took a little
What?
micro pipette, the gel, God thing.
The blood type.
What was that? What was that? What was that DNA?
DNA type.
Blood type.
Genotype.
Blood typing, right?
Oh, what?
It was genotyping. That's called.
Genotyping is different. Genotyping
Well, then it was
is different.
just
What is DNA gel?
DNA.
I
It
am
just
talking
tells
about
you
the DNA gel. So you have run this gel like that and
Yeah,
you can
and-
create the bands.
I don't know, it's
Yeah, agarose gel.
There you go.
Agarose gel. But what was the DNA?
Oh,
We did like
oh,
a purge.
I think you use the plasmid. If you did here, then we are using the plasmid.
I did.
Plasmid DNA. What is plasmid?
What?
Or not
What is
like-
plasmid?
No.
It's like fluid.
Plasmid is the eukaryotic, that is E. coli DNA, cytoplasmic DNA, that is called plasmid. In the plasmid, we can incorporate our DNA into that. Okay? Suppose you need to produce insulin in test tube. How can you do that? You can take out insulin gene from human, you can put that into bacteria plasmid, and then you can culture the bacteria so you can produce insulin into the solution. Okay? That is called DNA recombinant technology. Using that one, then I can take out that DNA from there. Then that is very small amount. How much do you know? 1 femtogram. Or suppose 1 picogram. Picogram means 1,000 times of microgram. That is 1,000, not 1,000 times, 1,000 times of microgram. 1 picogram of DNA, then if you run that into the gel, you will not find any band. So what you need to do, you need to make the copies of the DNA in your test tube. Making the copies of DNA, that is called the PCR or polymerase chain reaction. And during your DNA preparation, you already did that PCR part, but I don't know whether it was not told or something. So these are the things during that time, just let me know, just send me, you are free to say all those things. If you need the DNA replication to be discussed a little bit more, or the transcription, if it is not clear for you, then just let me know. Send me one email that I cannot understand this part, then I will go through all those things again. Okay? A little bit, and a very short way. It will not be difficult. Yes. Because I made the translation, it is little shorter. Okay? Okay. So there is a chance that we will not meet anymore. And there is a chance that we can meet on April 13th here. And between that time, we'll be online. And just if you are interested into doing the volunteer with me, just before going for the original classes, I'll make the thing, just let me know then, because I need to connect with somebody that who can see that, okay, I can see the slides, I can see this whiteboard. So I can just check with the whole system is on, then you can go for the classes. Class time will be same, 11 to 12:20. And what you need to do, just sit at your home, take some coffees or something, and log on into the computer and turn it on. That's it. Yes. Probably not. Probably not. You need not to be that is official dress or something like that, because the videos will not be on. Generally, the video means the PowerPoint slides and the whiteboard. Only that we need not to see the face to face. Probably the voice will be on, and I don't know whether the voice will be on for the both sides or not. Because when I did Wave EX one time, that voice is only for the instructor, the voice was on, and the students can type their questions there. Okay? So I'm thinking about in such way that you can just type the questions there and we can answer and we can have the discussion. Everything will be there. Similar like class, just we cannot see each other.
I guess it's a conference.
Yeah.
But, would this link be on Canvas or?
Yeah. There is a conference in Canvas. I am trying to use that conference. So actually, I was trying last night, but I didn't find anybody to connect to it. So I was trying by myself. That was, and I need to connect with somebody to see that how can I show.
I'll volunteer. I'll volunteer.
That is for the normal class times. Normal class times. I need to finish the course and you need to finish the course. So our target is something like that. We have to finish the course anyway, both of us. And rather than just meeting with each other, we will do the conference in that way. Okay, the same thing will be there. All the class format, everything will be there, but we will be doing the conference in such way. Okay? But the main thing is that you can drink some coffee, you can drink some foods there. Even you can lie on the bed to watch these things. So that is fine. You can sleep, nobody will see that.
Do we have to watch it while it's like being broadcasted, or can we watch it?
I don't think it is not be on. It is live. It is live. It is totally live. Because we don't have that kind of situation that we can create the video, we can post that one. Because we are in what situation? I can tell you. Just within 1 day notice, we have to move everything from lecture to online. It is not possible for that. Because online, we need a little bit of modification. Because even for my lab, I am preparing, I am bringing my camcorder to record the videos of how to prepare the DNA gel, how to prepare the spectrophotometric things. I have to do that one within 1 or 2 days.
Oh, Lord.
And it is very difficult to continue the labs. It's not really
Yeah.
easy. You cannot do that. So this is something like that.
In a month, why not?
I just got an invitation.
You just got a pretty
You got the invitation?
We're getting alerts, oh God.
So they just have to click start,
Did you just like
Yeah.
invite us?
just everyone in the class. So if you click start,
Web conference.
Invitation.
so you just have to click
Oh.
add conference.
Yeah.
And then, I don't wanna press start, because I might start a conference or something.
Yeah, I did by myself one.
Like,
Connect
I was like,
What I did was, I pressed conference, like, press select all members in the group or in the class. And I think if you.
Okay, there is one vital thing I need to talk about. The first thing is that make your group project ready, connect with each other through that is Canvas, and make your group project ready. If you have some kind of question, just discuss with me into that part. That already the group is there and discussion is on, so you can discuss everything there. I don't know what will be the presentation format. If we are in the class, the presentation will be here. If we are not in the class, the presentation will be through Canvas.
Okay.
Okay, so you have to present on that day. That day the videos will be on. Okay,
First of all, we have to figure out how that works.
that day the videos will be on. And we'll be matured until that day, I think, about Canvas.
In the middle of the woods.
Okay. We'll early today.
This is our first.
Dude, you don't